Okay, thank you for waiting till this main event of today's webinar. Now I'm gonna connect with Professor Lucas for today's main event. Exclusively for this webinar, here we have invited Professor of Neuroscience at Manchester University. Well, is Professor Lucas there? Please join me welcoming him. Hi, Rob. How are you? How are you today?
Hi, Elio. I'm very well, thanks. And you?
Okay. When I had a lot of discussions with my lighting customers, I got so many questions relating to our human-centric lighting offerings. And then I collected some fundamental questions as well as some tricky questions that I had always difficulty answering. And I am also very curious about, the deep background of this whole thing. So I think today we have a great opportunity to talk directly with Dr. Robert Lucas on these interesting topics. So I'm gonna show you the question first, and then we'll take this opportunity to get Robert's answer as a short lecture format after each questions. And at the end of the session, we'll have more free Q&A sessions. Okay, I'm gonna start with the first question. Rob, your paper in the circadian entrainment study measuring and using light in the melanopsin age has been cited by 814 other literatures. Actually, I Googled it. And I was also amazed by seeing that your another paper, melanopsin and rod-cone photoreceptive systems account for all major accessory visual functions in mice. It's been cited as by as many as 1,178 other researchers. What do you think is the reason this topic became so popular?
Thanks very much for the questions, Elio. And I just start out by saying what a pleasure it is to be here, and thanks everyone for listening in. So I think there are two sort of separate reasons for why this topic has captured people's imagination and led to so much additional work. And one of them is about just the science, and it's a cool bit of science, and the other is how important it is and the kind of practical relevance. So what I thought I'd do to start with is take us through those two kind of ways in which we can think about this topic. Mm-hmm. So I'll start out by talking people through the science and give you a flavor for scientifically why it's been such an interesting discovery. And one of the reasons is that it relates to a part of our body, the eye, that we actually understand in terms of its function and how it performs its function at a really very, very advanced level compared to lots of other things, lots of other parts of our body. So the conventional way in which the eye is supposed to work is that light comes in from the front of the eye here, of course, and is focused to form an image on this very, very thin tissue at the back here called the retina. And the retina is actually an extension of the brain. It's neural tissue there, which then performs the really interesting task of detecting that light and turning the flow of photons into a biological signal, which can then be sent to the brain. And that is supposed to be achieved by this group of neurons here. So the first step then in vision is light absorption by these cells, rod and cone photoreceptors, which are the last cells actually in the retina that the light falls on. And they absorb that light and translate it into a kind of electrochemical signal, which is transmitted then through other layers, other neurons in the retina, which for today we needn't worry about, before reaching these neurons here, the retinal ganglion cells. And those retinal ganglion cells you can think of as the output cells of the retina. So they have long processes that bundle together to form a nerve nerve bundle, which acts like a cable, if you like, an electrical cable, and it leaves the eye and it sends that information to the brain. So the very sort of the standard way that the eye is supposed to work is that the light is absorbed by rods and cones, translate that into a biological signal, which reaches the retinal ganglion cells, and the ganglion cells send that information to the brain. And in the human eye, there may be somewhat more, north of 1 million retinal ganglion cells. And that is how almost all of those retinal ganglion cells work. It's only a tiny fraction of them, much less than 1%, that do things totally differently. And what they do is they are able to absorb light themselves and generate their own signal, which they can send to the brain. And those few thousand retinal ganglion cells are called intrinsically photosensitive retinal ganglion cells, or IPRGCs. So that's the kind of science behind it, and why it appeals to people, because this is something that we thought we understood very well, and yet there's a whole other way in which it can work, and it highlights the need to understand things in great detail. Because if you were to say, how does the eye work? 99% it works like this, but that doesn't mean it's the only thing it can do. That would be interesting in itself. But it's particularly interesting when we consider what these IPRGCs do. And actually, the list of things they do grows the longer we study them. But the main thing that they do is they're responsible for measuring the daily change in ambient light intensity between night and day across dawn and dusk. Sounds like quite a simple task. It's incredibly important for our biology, because we, of course, had very, very profound 24-hour rhythms in our physiology and behavior. So we can, the most obvious instance of this is our sleep-wake cycle. So almost everyone sleeps at night and is awake during the day. But there are almost all aspects of our biology has this 24-hour variation. Try having a glass of wine for breakfast. Has a totally different effect than it would if you have it for dinner, for example, and on and on and on. So we have these 24-hour variations, and obviously they're a reflection of the Earth's rotation in the 24-hour external day. So how do IPRGCs fit into this? Well, IPRGCs are central to establishing those 24-hour variations in two ways. One way is that we have an internal biological clock, a circadian clock in our brain, which keeps a 24-hour rhythm and drives many of these changes in our biology over the day. But like any clock, it's only useful if you can set it to the right external time. And it's IPRGCs measuring that change in light intensity between day and night, which is responsible for setting the phase of our biological clock. When you experience jet lag, it's IPRGCs which are dragging your clock back to the correct time, or pushing it forward to the correct time, depending on where you've gone to, so that now over time you get resynchronized to your new local time. So that's one thing that they do. The other thing that IPRGCs do is they can provide a direct kind of gain control of many of these daily rhythms. So, for example, our propensity to feel sleepy, our alertness, this is controlled by light as regulated by IPRGCs. So in a very real sense, those IPRGCs determine when we feel sleepy, when we feel alert, when we feel hungry, when we're able to metabolize food well, when we respond well to sugar, when we want to use the toilet, when we have most severe symptoms of many diseases, when we're best to perform athletic and academic tasks, and on and on and on. So this is a tiny number of cells in our body that has a huge importance for our general health and well-being.
Well, thank you, Rob, for your comprehensive answer to the first question. I think the next question goes to why actually this most important discoveries on circadian entrainment by IPRGC in the retina and the subsequent influence on the hormone melatonin seem to have just taken place right before and after the year 2000. What happened then, and what made you a pioneer in the research?
Yeah, Elliot, it was really such an exciting time. A few years then, when it just seemed like every year there was another big advance in our field, coming by no means only from my own work, but from many other researchers. And to take you through that and maybe give you a sense of the development of this field, just want to take you back to that time. And what did we know in the run-up to this period, as you say, around the year 2000? And the sort of the framework for it was reports over the previous 10 or 15 years from humans, but also from other animals, that you could have blind people that still had some response to light. And to give you an example of this, I've just taken this figure from a paper from Chuck Seisler, who's at Harvard, who published this in 1995, a really important paper, looking at that hormone you mentioned previously, melatonin, which is produced at nighttime. So I'll take you through what you can see here on this trace is that we have throughout the daytime, melatonin production is low, and then over the nighttime it sort of rises and it peaks. So you can see, if we start at the left-hand time, you can see we're marching over 48 hours for this person in the lab, in Chuck's lab. And you can see that melatonin starts low, and then it gets high as it goes through the first night, and then it falls away again, and then it gets low again, and then it rises again on the second night. And then you can see about 2/3 of the way through that second night, there's a period when the lights are turned on bright. And those bright lights have the effect of suppressing melatonin. And you can see that very effectively, that the melatonin level goes right down. What's very interesting about this trace is it's from a subject who couldn't see anything. So this is a blind person who still has this ability to respond to light in a totally subconscious way. So that was the kind of background. And my own work in the field started when I was working with Russell Foster, and we were interested in looking at that phenomenon in more detail, and particularly how we could get at it by using the tools that were then becoming available in laboratory mice. And what we could do in lab mice was to use the tools of genetic engineering to change the mice in very, very defined ways, and particularly to generate mice that didn't have rod and cone photoreceptors. So remember, at the time, everyone would have assumed that rods and cones are responsible for absorbing light. So if you take away the rods and cones, then we should have an animal that was totally blind, totally incapable of responding to light. So when we did that, the mice were actually visually blind, but they were able to still show circadian responses to light. So indicating again here that there must be something else other than rods and cones which are responsible for these important light responses. So that tells us then that there is somewhere a new photoreceptor which is providing these kind of circadian light responses. It posed to us, to the world, a couple of really important questions. First of all, where is it and how does it work? But in a way equally important from a practical perspective is what sort of kinds of light does it respond to? And actually, we first set out answering the second of those questions because we felt that this was an important way of understanding this new photoreceptor, which didn't rely on us first knowing actually what it was. So one of the things we wanted to ask, of course, is what wavelengths it's responsive to, which for this audience, you will well understand why that's important. A challenge of doing that with the circadian system is that every time you present a light stimulus and wait to measure a response, which you'd like to do, right? Present a light of a given wavelength, wait and measure the response, use a different wavelength, wait and measure the response. Each response takes several days or weeks to measure. So it's very, very long-winded if you want to try different wavelengths. So we wondered whether there was something more immediate, a more immediate light response that we could generate from our rodless coneless mice that would allow us to do this in a more kind of efficient way. And so back in 2001, we wondered whether actually a different sort of light response, the pupil light reflex, might be regulated by our new photoreceptor in these mice. I'm going to show you a video here. So this is a close-up of a mouse's eye. Looks very much like our own, right? Or at least certainly like any of your pets at home. And you can see around the outside that it has this clear the iris, and the middle is dark where the pupil is. So this is a mouse in the dark, and the pupil is nice and big, as it would be for us. So as we play the video, in a moment you're going to see a flash of light there, and you can see that this pupil constricts. Right, there's a big pupil constriction, and then slowly it redilates over time. So this is still one of the most exciting things I've seen in science. We had no idea that it was going to work, but actually when you did, you can see it's just, you don't even need to measure it. It's so obvious, right? So it's a very, very cool thing. And now, of course, the pupil light reflex is used in a lot of situations to measure this new photoreceptor activity in humans. But at the time, we had no idea that it was going to work. When we had this pupil light response, we can now then ask what wavelengths of light most effectively elicit that pupil response. And so this is the paper we published on that, one of the figures from it, showing how how sensitivity varies as a function of wavelength for these mice, the black rods and cones. And you can see that there's a peak just shorter than 500 nanometers, and actually it's at 480 nanometers is the most efficient wavelength, but broad sensitivity around that kind of cyan part of the spectrum works well. And so we concluded that this new photoreceptor was maximally sensitive around 480 nanometers, and particularly that is to the cyan part of the spectrum. We also looked at how much light you needed to give. I won't take you through that in detail, but interestingly, it seemed like light intensities that correspond to around the twilight to daylight range is what our new photoreceptors were responsible for measuring, which of course makes a lot of sense if you're thinking about tracking those day-night differences. So later on in 2001, something else really exciting happened for us, which was that the labs of Bud Brainerd and Deborah Skeen and Joe Arendt published an action spectrum for the suppression of melatonin by light. So that ability of light to suppress melatonin in humans as a function of wavelength. So really heroic efforts and huge amount of data collected by these guys. And they found also that in humans, that cyan part of the spectrum was especially effective at suppressing melatonin in humans. So these two bits of information come together to identify cyan as a really important part of the spectrum for these sort of circadian light responses. Okay, what about the other question? So the other thing that we'd really like to know is, what is this photoreceptor and how does it work? And there have been two kind of strands here to bring together, one of which comes from the work of Iggy Provencio and Mark Rolag, who were busy studying this animal here, Xenopus, which is the African clawed toad. So it's a type of amphibian, a toad. And they were interested in how the skin of Xenopus changes its color as a factor of background light intensity. And it does that, so it gets more or less pigmented depending upon how much light it's exposed to as a kind of a camouflage. And they identified a protein called melanopsin, which they thought was important in that process, and went on to show, surprisingly perhaps, that melanopsin is also found in humans and other mammals. So we don't have the same change in pigmentation of our skin that Xenopus do, but we still have this protein in our genome. And we'll come to that in a second. Another really important breakthrough was made by David Burson and his group, who were studying the retina of these guys, rats, and they were recording the activity of retinal ganglion cells. And they found a very few retinal ganglion cells that were able to respond to light, even if there were no rod and cone photoreceptors present. So directly sensitive retinal ganglion cells, for which the term IPRGCs, intrinsically photosensitive retinal ganglion cells, was coined. And work from Samahata and Kim-Wai Yao showed that those IPRGCs contain melanopsin. So now we bring together a link between a protein which is responsible for light sensitivity in frog skin and these light-sensitive retinal ganglion cells. And over the next few years, work from my own lab and also many others around the world showed that deleting melanopsin in mice leads those IPRGCs to lose photosensitivity and the mice become less sensitive. If you take melanopsin and put it in a cell ordinary cell, any cell that is not light sensitive, like most cells in our body, they become photosensitive. So you can build photoreceptors just simply by including melanopsin. And finally, if you get rid of those IPRGCs in a mouse using genetic engineering, you can generate an animal that can see just fine, but can't tell night from day. And that brings the whole thing round in a way over those few years, from starting out with a hint that there might be something there that, frankly, people who understood the retina or understood vision thought was a crazy idea that there'd be a photoreceptor other than rods and cones, all the way through to really establishing what this photoreceptor was, the kinds of things it can do, the wavelengths to which it's most responsive, and the way in which it supports our physiology and behavior.
Well, it looks like those years were really exciting periods for the people in that area study. I have a little question here. When you talked about the engineered mice, and those engineered mice lack rods and cones, and in the most recent years, you eliminate IPRGC from mice. I think you mentioned like some genetic engineering there. Of course, you cannot really eliminate, the IPRGC just one by one with faucet or something like that. So could you explain a little more details about the genetic engineering that you made use of in order to control your experiments?
Yeah, so it's one of the things that you can do in mice that obviously you can't do in humans, for example, is you can take their genome, so the particular, go into a very particular part of the genome and change it in any way you like. So, for example, melanopsin knockout animal, to delete melanopsin, you can go to the melanopsin gene in your genome and just change it so that it can't work anymore. That's the simplest way that you can do it. And then you end up in an animal that all of the other genes are completely fine, everything else works completely fine. The only thing it can't do is produce melanopsin. So in a way, you can think about it in terms of targeted introduction of kind of the type of genetic variation that occurs randomly in nature. So all the time things are getting broken in our genome, mostly they don't have really much of an effect. Well, what we can do is to say, right, we don't want a random change in the genome, we want to change a very particular part of it, and you can go in and do that. Then the other thing that you can do is you can say, right, this is a part of the genome which is responsible for producing melanopsin. Right? So now what I'm going to do is I'm going to change it so instead of producing melanopsin, it produces something else. And that something else could be something that makes the cells die. So wherever the melanopsin would ordinarily produce, instead of producing melanopsin, it produces a toxin which kills that cell. And then you end up with an animal which has these IPRGCs, where melanopsin is expressed, eliminated.
Wow. That's fascinating. And I think there was a very brilliant discovery of the melanopsin in the skin of the Xenopus, and the same type of melanopsin in human eyes. So I guess there must be some linkage between the amphibian and humans, one of the mammalian. Is there any evolutional point of view that you can add some comment? With some comment?
Yeah, thanks. It's a fascinating question and a fascinating topic for discussion. So if you think about this Xenopus, let's consider the Xenopus. You can see it's got eyes, and in its eyes it's got a retina, and in its retina it has rod and cone photoreceptors, the same that we do, using the same proteins, very, very similar really to our own rod and cone photoreceptors. It also has melanopsin in its eyes. Actually, it's got two sorts of melanopsin. It's a complication we don't necessarily need to go into. It then has obviously melanopsin in its skin. It has also photoreceptors elsewhere in its brain, and its pineal gland is light sensitive. and it has light-sensitive muscles. All of the muscles in its body are light-sensitive. So are its major organs, its heart, its liver. And all of that allows almost every single cell in the body of that organism to track the daily change in light intensity. And it does that not only from melanopsin, but actually from a lot of photosensitive proteins. What has happened in the evolution from frogs to humans is that we've lost almost all of that light sensitivity. So we only have light sensitivity in our retina. And of all of the different ways in which that Xenopus has of detecting light, we've only really kept 3. So we've kept the rod photoreceptors, we've kept the cone photoreceptors, and we've kept melanopsin. We've ditched all the other things. Why that is, who knows? But it does mean that fortunately, we as humans are much more simple in terms of photoreception than Xenopus. So if we think that the challenge of lighting engineering for circadian health is complicated for humans, just as well we don't have to do it for Xenopus.
Oh, wow, that's an interesting story. If we had all the light sensitivity all around our skin, our lighting engineering must have been a lot more difficult than it is now.
Yeah.
Yeah.
Or more fun, depending on how you want to look at it.
Right. Maybe we can go over to the next question. The discovery of iPRGC and the melanopic action spectrum are such a great achievement in the physiology and neuroscience. However, other than the academic advancement, do you also expect a lot of practical benefit of lighting that are tailored to improve human productivity and health and well-being? What do you think?
So I think to answer that question, I go back to the slide I showed at the beginning, really. We're talking about the significance of iPRGCs, the type of ways in which they influence our biology. And this is not an exhaustive list, but you can see there's a lot on it. So the capacity for iPRGCs and melanopsin to influence our biology is really very, very extensive. And I think from the perspective of translating the science to lighting practice, that's probably one of the most important things, is the kind of appreciation that lighting is not just to support vision or to create a pleasing effect. It's not an optional extra. We really do have to consider how our lighting choices impact on our biological rhythms and sleep patterns, etc. So understanding the very sort of fundamental importance of this system immediately, I think it pushes us to take that into consideration when we're thinking about the light that we expose ourselves to and our pattern of light exposure. The other thing that I think the fundamental understanding of the biology helps us to do is, of course, if you understand a system at a mechanistic level in terms of principles, you can start to think about how it is you'd like to manipulate it. And one of the most important questions is to ask, what sort of light do we need during the day, and what sort of light do we need at night? You know, what is the right sort of light exposure? And I believe you mentioned earlier that the Wells standard, which has some guidance on this. We in Manchester, about 18 months or so ago, had an expert workshop from scientists around the world who consider this problem based on their sort of research activity, and came up with a sort of a consensus proposal, which is published in this paper here, which there's the DOI for, people can have a look at, about what we should be heading for, what represents a good amount of light during the day and at night. And that's our quantitative guidance there in terms of melanopic EDI, ideally greater than or equal to 250 lux during the day, and during the evening less than 10 lux, and during sleep less than 1 lux. That's what we proposed. So that's kind of what we think, based on the balance of science at the moment, is the kind of thing that you should really be heading for, at least under for regular sleep-wake cycles and regular situations. And of course, it's worth pointing out that this is not achieved under many circumstances at present. So this is just one study from our own group, but there's many others from others around the world. So this is a recording of light levels at the intensive care unit at our local hospital. And the patients in that unit are both exposed to less light than you'd like during the day, and certainly more light than you'd like during the nighttime. So if we can use that knowledge and try to hit some of these targets, then there certainly is scope for pushing our light environments more towards what we think is necessary for a healthy biology.
Well, as I learn more about the science behind this human-centric lighting and circadian rhythm, I become more and more committed to making some difference in the real lighting industry. I appreciate this opportunity very much. Here we turn to a little more practical questions. The next question is about how we implement the best human-centric lighting. The lighting designers are really fascinated by the new possibilities of functional lighting, exploiting the spectral tuning. However, at the same time, overwhelmed by so many new jargons and metrics, such as MP ratio, EML, MDL, and melanopic EDI, and circadian light, and circadian stimulus, circadian potential function even. So what's your recommendation to a beginner lighting designer who is being confused and overwhelmed by all these metrics? Can you suggest a simple ABC steps?
So I think that what I'd like to do is to start just by giving you an overview of how these various different approaches vary, because I think that's quite important. So as you said in your introductory presentation, the fundamental problem we have here is how you can go from a spectral power distribution here, in which energy varies as a function wavelength, and collapse that into a single number which describes light in a sensible way. And of course, for illuminance and for lumens, we use V lambda, which is the photopic sensitivity function. That's the curve here in the red, where you multiply the spectral power distribution by the red line, integrate across wavelength, and that gives you a number. So the fundamental problem that we face for the melanopic circadian side of things is, first of all, to define what our function looks like, what our kind of normalization function looks like, weighting function looks like. So if it's not the red line, what is it? And it's going to be something like the blue line, right? Which, as you say, peaks more in this high-end part of the spectrum. So the various solutions that are out there vary in terms of the specific form of that blue line in more or less significant ways. They can also vary in the scaling factor. So how big is the blue line versus the red line, or alternatively, once you've integrated, once you've got your number for your non-visual light response intensity, how do you apply a scaling factor to make it equivalent to photopic lux or whatever you want to do? And in terms of normalization and processing, so as you say, there's an argument for looking at MP ratios where you take your melanopic radiance and your photopic radiance, and then you can have a good way of comparing them. So without going through each of them in detail, I think the simplest thing to say is that there's only one for which there's an established SI compliant international standard, and that was codified by the CIE in SO26. So I was a member of the committee that performed that work. And that defines a blue line, which we call the melanopic spectral efficiency function, right? So that sets the form of that that blue line, if you like, and also provides a variety of ways in which suggestions in terms of how it's scaled with respect to the other ways in which you can calculate light and the ways that you can process it. So it's also the route that's really most widely used by the scientific community. So I think whatever you start with, to be future-proof, you should be using that CIE standard and the melanopic spectral efficiency function. Okay. So then the second thing to think about is, what is it that we are trying to establish? And again, as you said nicely in your introductory presentation, what's relevant is how much light falls on my eye when I'm sitting, standing, occupying that environment. And so for that, we need to measure melanopic irradiance at the subject's eye. Okay, so that's the simple thing to do. It's a little bit, the problem with that number in a way is it's a bit hard to relate to what we already know about the photopic illuminance, because there's a different sort of number, and also to think about to have kind of an intuitive feel for what that number means compared to outside daylight and so forth. And so as a result of that, as part of the CIE, there's a suggestion that people could use the melanopic equivalent daylight illuminance, for the melanopic EDI, units are lux. And that is the illuminance of daylight, or in CIE terms, the D65 light source, that is required to give an equivalent melanopic irradiance. And I think that is the most kind of intuitive way of describing this melanopic irradiance from one situation to another. So I would recommend people use that when you're considering what the people occupying the space are going to be exposed to. And there's an Excel toolbox there which people can download, in which if you include your spectral power distribution, it'll give you your melanopic equivalent daylight illuminance relatively easily. As that standard has more life, I hope that there will be like an ecosystem around it will develop in terms of a meter that will allow you just to measure that. There are some things around and about. I don't know enough really at the moment to recommend any of the solutions that are there over any of the others. And maybe it'll come sort of be incorporated in the kind of the software that you guys used when you're making engineering decisions. So that's the route I think that people should be looking at. They should be considering this melanopic equivalent daylight illuminance. And how you can achieve that.
Well, thank you very much for the detailed description of how we handle those metrics and how we calculate the important metrics of circadian lighting. Well, I have a recommendation for the audience, for those who are really interested in calculating the melanopic EDI by yourself. I recommend that you start with the given toolbox. Get your hands on first and take any spectral power distribution data and plug that in, and you can immediately see that how much photopic lux converts to MEDI. And the second step I would recommend is that you start with the formula. And then it's not so difficult. You can still use Excel to calculate the melanopic irradiance or any alpha opic irradiance, and then you go through the normalization process, which is the actually very confusing part. The normalization can be done in a lot of different ways. However, you must go through that difficulty once. Then you should be able to double check whether your answer is in coincidence with the toolbox answer. Then you'll be more confident in taking care of all these metrics and should be very confident in designing your lighting system. And all right. Okay, good. All right, then let's move on to the next question. All right, the questions are going to be a little bit trickier, more and more. Can a cyan-rich daytime lighting really improve people's concentration productivity? Well, we've seen a lot of evidence so far. Well, we observed such a result from a clinical test. Indeed, what other benefits can we also anticipate? Does high MEDI or MDEI, MDEI is like kind of typo of MEDI, light exposure during daytime also help better sleep at night? So is there something like melatonin rebound by having a bright sunlight or, high MEDI light during daytime helps better sleep at night? And if so, could you explain the physiological process behind that?
Okay, let me take those two questions separately. First of all, whether lighting can improve concentration and alertness. So I think if you look at the scientific literature as a whole, there are both field studies, the sort of things that, or at least pseudo field studies, the type of stuff that you presented here, and also lab studies showing that light can have this effect on alertness and concentration. So there's definitely scope for light to have this effect. There are also, of course, instances in the literature where people fail to find an effect by changing the light. Well, there may be many reasons for that, but I think one of the important things to keep in mind when we are thinking about these kind of effects is that although light can influence alertness and concentration, so do many other things, right? So we know the time of day will, so that's another influence on light and alertness. How well you slept the night before. So this is me on the night that I let my dog sleep in bed with me. I'd strongly recommend against that if you want a good night's sleep. After that, sleeping with my dog for the night, I was not very alert the next day. So again, the sleep you had the night before, of course, caffeine consumption, on and on, how interested you are in the task, all the other things that are going on. There's multiple things that will impact upon something like alertness and concentration, and that makes it hard for any individual person on any individual occasion to work out how much light is going to be influencing that parameter. So the capacity is for it to do that is there. How much it does for any individual person on any individual occasion is of course going to be contingent on all of these other things. So then the other question about whether bright daytime light enhances subsequent sleep, I think this is a topic that's actually we're working on this at the moment in the lab, and it's something that I'm very interested in. And I think at the moment there are some things that we can say, and other things where there are kind of hints that it's going in this direction, but probably a bit too early to really stick our necks out and say this is definitely happening, right? So let's start with the effect that I think there is sound evidence for from a number of directions, and that is that exposing yourself to bright light during the day can help you to have a more appropriately phased biological rhythm with respect to the light-dark cycle. So for many people, that would be going to bed earlier and sleeping longer. And just to introduce you to that, I want to show some data from Till Roneberg and his colleagues in Munich. And they've really pioneered this idea of social jet lag to describe the relationship between our sleep patterns and clock time under most circumstances. So this image here is a sleep diary for someone over 6 weeks. And what they're doing is they're noting when they go to bed and when they wake up. And that's the red and white hash bars or the green bars here on day after day. So it's clock time along the bottom, you can see it highlighted there along the top. And you can see this is a person that sleeps during the nighttime, which is great. So what's different between the red and white bars and the green bars? The red and white bars are work days and the green bars are weekends. And you can immediately see that this is a quite typical sleep pattern where during the week, there's very little variation in when this person gets up, right? They're always getting up or invariably getting up around 7 o'clock in the morning. And that, of course, is because that's when the alarm clock goes off and they have to go to work. On the weekend, the alarm clock doesn't go off, and so they can sleep in, and they sleep in 10 o'clock, something like that, on the weekend. What that tells us is that during the week, this person is getting up earlier than they would like to get up based on their internal biological clock. And as a result, they're relying on an alarm clock. So how Till describes it is in terms of social jet lag. So this person is really adapted to a time when the sun should be getting up later, to working at a slightly later time zone, which you can see is what happens on weekends when they stay up a bit later and then sleep in. So this has a number of effects. The first thing is, because that alarm clock is waking them, it tells us that this person is waking up too early. Their clock should be allowing them to have, their circadian clock, should be allowing them to have more sleep. And indeed, what happens is that throughout the working week, people don't have enough sleep, so they have reduced sleep, and then they catch up on sleep on the weekend. The obvious solution is to go to bed earlier, right, and get sleep and get more sleep that way. And bright light during the day, in both field studies and lab studies, has that capacity to shift the clock so that people are more inclined to go to sleep earlier, and therefore have a longer time to sleep before they wake up on the next day. Either because of either they'll wake naturally, or the alarm clock wakes them later in their sleep episode. So that's one important way in which having brighter light during the day can help. The other ways are more kind of, as I say, more sort of straws in the wind possibilities than definitely defined phenomena, I would say. And one of those you mentioned is the possibility that we might have enhanced amplitude of biological rhythms under a light cycle. So this is just one data set. There's a small number of papers looking at this. And this is data from Mishima and colleagues back in 2001, where they're looking at melatonin production from elderly people with sleep problems. And the lines to keep an eye on here are the squares. The line with the square symbols, that's the melatonin production of these guys when they're under sort of ordinary light exposure. And then the circles are the melatonin production for these subjects when they're exposed to enhanced additional bright light during the day. And you can see that there's quite a big difference here in the amplitude of that melatonin rhythm. And there are a couple of papers showing that. So there's some indication that that's the case. That's the case. We're currently working on that to look at that as a sort of an independent biological phenomenon, doing some experiments in the lab, and you know, it looks as if this is a genuine thing. But I think that that's something which we really need to see developing over the coming years. But it certainly is an exciting possibility.
Well, it seems like all these research results are talking about the general old wisdom of having good outdoor activities during daytime will give us better health and more regular lifestyle. It seems like the person with social jet lag is torturing himself or herself in a way that the person is flying over from, in case of in the US, from West Coast to the East Coast, and from East Coast to the West Coast every week. It seems something like that. And also, yeah. So, okay, go ahead, please.
Sorry, carry on. No, well, I think it's more that they are living in New York, but on Los Angeles time.
Mm-hmm. Right.
So you know, that's more the situation. And if you think about if I put myself back to my teenage self, for example, roll back the years, this is very much how I felt, right? That I was not sleepy at nighttime, mm-hmm. and I would struggle to go to sleep. But then, when that alarm clock woke me up in the morning to go to school, it was hell. You know, so these are the sorts of things where lighting could make a difference during the day.
With all this enlightening knowledge about the human biology and physiology, we could somehow make ourselves healthier by making our lifestyle more simpler in a way that we get bright light during daytime and dim light during nighttime, and maybe even getting more help from specially tuned lighting conditions. However, for ordinary healthy people, they have the freedom to move in and out of their house or their workplaces. However, there are people with disabilities or patients who have limited, mobility and capability to move in and out of the building during daytime. So, for those people who are having difficulty getting enough bright light during daytime and adequate dim light during nighttime, I think it would be a great improvement for their health and well-being to have a right lighting environment, right?
It's definitely a more important consideration. Well, definitely. My view is that it's a more important consideration for people who don't have that level of control of their light exposure that you and I might have in our ordinary days. I agree with that because, if I regard it from my perspective as a sort of research expert in this field, one of my most important things that I should do really is just explain to people what they can do with their own lifestyle, right? And for many people, that's a very important message. What can I do myself? Go for a walk in the morning, be exposed to the natural light cycle. Sunlight is best. So that's an easy sort of answer. But as you rightly say, we also need to think about those people who don't have that same level of choice.
Okay. Well, now I have a little more tricky question here. It's actually from one of my colleagues. Which do you think is more adequate choice of street or road lighting? A high MP ratio, high MEDI lighting that prevents accidents, or low MP ratio or low MEDI lighting that does not disrupt the driver's natural secretion of melatonin? And pretty much the same question may go to nighttime workers and shift workers. Would they be better off by shifting their circadian rhythm to their work hours, or by keeping their original rhythm by minimizing the light-induced phase shifting of their circadian rhythm?
Right. So you know that I'm not going to answer those questions, don't you?
That's why I said it was going to be a tricky question.
It's very, these then have become, there is no simple answer to those two questions, right? And I'll take a few slides just to explain why. And it starts really, even if we just think about these kind of circadian or non-visual aspects of light, the positive and negative sides of that light exposure are two sides of the same coin, right? So if you want light, if we think that light can drive alertness, then it follows that it can disrupt sleep. If we think that light can provide stable biological rhythms, it follows that if you give it in the wrong way, it can disrupt them. So, we're always balancing the positive and negative effects of light, and that in itself is an important decision in terms of these circadian and non-visual effects. On top of which, of course, we're using light to avoid hazards, to perform tasks, to enjoy our surroundings, to communicate with friends. All of those other things are out with that kind of, or outside that, those kind of melanopsin circadian effects of light, but they're equally important, right? Or can be very important. So deciding which of those, the relative importance of these various things, of course, has to be done on a case-by-case basis to a certain extent. And I think it's the way that I think about it is in terms of a target, right? Understanding the melanopsin system can help us to hit a target, because we can start thinking about what it is, what do we need to do with our light exposure to hit that target that we've asked for, right? But determining what target to hit is, of course, a much more complicated task. So on the one hand, we might be trading melanopic effects against other considerations. So, for example, that would be the situation potentially for your street lighting. So the melanopic, if you make the street lighting too bright, then it's going to disrupt sleep and disrupt circadian rhythms, right? On the other hand, if you make it too dim, maybe people are going to have to will start having accidents or feel unsafe and those sorts of things. And, you know, this is addressing the relative importance of these various impetus, these various kind of drivers is something that I'm afraid has to fall to someone other than myself, because it's all about for that person and that group of people in those circumstances, what do they value more and what's the relative importance. So unfortunately, maybe you can try to mitigate the negative effects on the circadian system by changing the lighting in some way, and that would be a useful thing to try to achieve. But at some point you come up to the point where you have to compromise, and that's a difficult one. The other thing that's difficult is to even understand what your preferred melanopic effect might be, right? So that was the instance where you're talking about the shift worker. Well, maybe as you come to the end of your shift, you want to use dim, low melanopic light to get ready so that it's not disrupting your sleep when you go home. Well, that might be fine if you're walking home or getting the bus home. On the other hand, if you're driving home, you really don't want to be sleepy, because then you could have an accident. So again, that's a decision that has to be made for an individual case-by-case basis, and probably beyond what we can talk about now. Mm-hmm. I don't want to leave it as on too negative a point, because I think where we are, though, is we can start saying things about what light exposure we're looking for under normal circumstances. When we try with 24-hour light, we're living a 24-hour schedule, and you know we have other constraints accounted for, and I think that's quite an important thing for most people under most circumstances.
Well, thank you very much for your very wise answers to those tricky questions. Well, right, there must be some priorities in each individual cases. We cannot really make a typical solution that's been used in common sense all over those different cases. Obviously, if you are a worker, working at night on a very dangerous task, I think securing your safety may be the first priority instead of keeping your original circadian rhythm. That's quite obvious. Well, I have a case where I definitely need awakening lighting. I think this may be implemented in some time in the near future. I mean, whenever I go to a classical music concert, usually the stages are lit by very warm, cozy lighting around like a 300 Kelvin, and the audience is in their seat without much lighting during the performance. And then the problem is that my wife and I paid a lot of money to buy the ticket, and then in the middle of the concert, I usually fall asleep. And I like the ambient lighting in those music halls, but at the same time, I like to keep myself awake all the way through the end of the concert. I mean, otherwise, I just get blamed from my wife. Why are you coming here to just take a nap? I mean, things like that. This is kind of an obvious case where we need a certain type of lighting. But again, there are really, really tricky cases where we have a lot of difficulty deciding which type of lighting is best for which circumstance, because it's not involving the decision from biology or the work performance, but also from the considerations of ethics and sociology. So,
Absolutely.
I mean, I have no intention to make the conclusion right here, today, but maybe this is the theme that the whole lighting industry should reflect on over the time. And more and more findings will come out, I think, from a lot of researchers.
Yeah, I mean, it would have to be a conversation between, the lighting industry and, well, in workplaces, the management and unions and people who understand the kind of sociology of it and the social science elements of it. So, if you wanted to shift people's clock for their shift work period, what happens on the weekend when they want to spend time with their family? Those sorts of things. It's very, very difficult to have one size fits all answers.
Right. Okay. This is all for the prepared questions for you today. That was fascinating. And I think we have now some time left to look over the questions that we got from the webinar system. There are a lot of the product-related questions, but one word just catch my eyes. I'm not sure how my wife will react when I start drinking wine at breakfast. But I'll tell her it was doctor recommendation. Thanks, Dr. Lucas. What is this? I have a little difficulty connecting this question to your lecture.
I recommend it under very careful
take an example of drinking wine at the breakfast, right?
No, a very carefully controlled experimental conditions. You have a single glass of wine for breakfast on one occasion, and you note the effects on your body compared to when you had it for evening. I'm not recommending it as a lifestyle choice, not by any means. You'll find that actually avoiding wine for breakfast is a good idea if you do it.
Yeah, of course, especially when you are expecting some serious work during the rest of the day, or if you are trying to drive. No, obviously it's not an option.
Absolutely. Yeah, don't do it.
Okay. What other questions do we have? Let's see. Which ones? Okay, let me pick this one. This is from Kenneth Martin, who I know actually in person. Which uncertainties are there on the linear scale on the melanopsin sensitivity curve? May this curve, I'm not seeing the whole sentence here. Okay, may this curve change in the near future its shape, even if perhaps the peak wavelength stays the same? Okay. Uncertainties on the linear scale on the melanopsin sensitivity curve. And then, do you expect any new consensus in the scholars in these areas of study and people, the experts from the industry? Is there going to be some possibility of consensus in the near future to adjust the curve a little bit in a way that it meets with some of new findings? Probably. I think that's the intention of this question.
Yeah. So I think it's really important to realize that any of these standards, they're imperfect, right? And they're never going to work for everyone all the time. The same is true of V lambda. If you talk to people like vision scientists, you'll find plenty of people who make the point that V lambda doesn't always work for aspects of brightness, right? Brightness perception. So, there's going to be differences between individual people is certainly an issue, right? So that inherent in that estimate that the melanopic function is a correction for lens absorption, right? And of course, the yellowness of our lens changes a lot between individuals and as we age. So immediately, as soon as you put down a curve like that, it's going to be wrong. It has to be wrong. Anybody's curve is wrong, whatever you do. So I think the issue really is, how wrong is it? Is it worth changing everything to make minor corrections, or is it as good, or is it performing a function as well as it can do? Given how recently it's been standardized, it's also been independently tested on published data, and it provides a realistic, reasonable correlation with published data which were not, those data were not, the melanopic function is not necessarily derived from those data, but it can predict those data, which I think is quite an important thing. I think for the moment it will be, it'll prove to be good enough for what we do. There certainly is uncertainty, as there is in everything here, in terms of exactly what form that curve should have. So, I haven't talked about the possibility that cone photoreceptors also contribute to these effects that we observe, which they certainly can do. And if you want to refine that, then potentially you can see that happening in future. But I think for the moment, we don't want. This is a good compromise. It's a simple solution. It has very few assumptions, and it works well enough to predict the data that we have at the moment, which I would argue it's worth sticking with until we really find something that is going to make a material difference, which for myself I think is unlikely.
I see. So there's no true melanopic sensitivity curve that represents individuals' response, right? It's wrong for everybody, actually. So it's the problem of averaging stuff. So
well, look at V lambda. It's wrong for everybody. It's not. There's, in some regard, there's nothing different about the melanopic function to V lambda. It's not right for me, definitely, but it's good enough to give an overview.
I see. So from your answer, my guess is that at least the CIE is not likely to change the current melanopic sensitivity in any near time frame.
So let me make it totally clear. I have no role with the CIE. So who knows what, as far as I know, they don't, but they might be. I sat on that committee just to help generate that standard, but I don't have any official role with CIE.
I see. I see.
It's my opinion, as somebody who works in the area, that this is a reasonable approximation of what of the challenge that we face, the real world challenge that we face of measuring light in a simple and reliable way.
Right. I think I agree with you on the point that on the average, the melanopic sensitivity curve works pretty well. And what we have to focus more on is that we should really implement the advancement of lighting onward, because so far, I've seen many people in the lighting industry who understand the mechanism and the importance of this, circadian entrainment by lighting. However, I face a lot of skepticism as well, whether about, people's willingness to pay extra money to, replace their existing lighting in their building, office, or school, or their home. So I think making the change is more important at the moment, because at least we all understand that the cyan portion of the lighting is the most important ingredient of the spectrum that affects the human body by suppressing the melatonin. And the little tiny difference might not be as important as the action that we actually take in order to make advancement in the lighting overall.
Certainly, we could spend a long time as scientists arguing about the exact form of that melanopic sensitivity function or whatever it might be, and that would be a delay in getting the message out to people that there's a particular light intensity that you should be shooting for under different circumstances. So I think there's certainly an argument for making that move to sort of take advantage of the science that we have, even though while acknowledging that nothing's perfect, that this is the best that we can currently do, rather than keep going around that circle. That's what I would say.
I see. Okay, thank you very much, Rob. Maybe I can still have just a little more time to pick one more question, probably. Let's see. Okay, let's pick the question from Markant. I know him in person as well. Thanks, Mark, for posting a question. So his question is, while we all understand that the skin of different races has, through evolution, changed to protect that race from sun exposure, do we think IPRGC's performance could be similarly different in different races? Do we think their performance changes with age? I think this question is closely related to the answers that you just gave.
So I think there is no suggestion. Let me say what I think would be a most unlikely change, and that is the spectral sensitivity of melanopsin, because it's the same in a mouse as it is in a monkey, as it is in a human, as it is in a rat. So it's most unlikely that cyan part of the spectrum is going to vary across the human population. Mm-hmm. It is true to say that we really need more information about different sorts of human populations and how they respond to light. I think that's a really fascinating area of science. There's recently a very interesting paper just asking how the sensitivity for melatonin suppression varies across people. And reporting, so this is work from Andrew Phillips in Australia, reporting really quite big differences in how much light it takes to suppress melatonin for different people. Oh. And understanding the origin of that variability is, again, something I think that for the field as a whole is a really important challenge, because it's going to because it could be that it's different people with different genetics or different inherent characteristics. It could be something about their environment which produces that variation. So at the moment, I don't know any suggestion that this change in sensitivity has a genetic origin, as kind of implied by the question of whether there would be differences in terms of people's kind of racial origin and so forth. But certainly understanding the origin of that variation, I think, is a very important question for our field.
All right. Thank you very much for your detailed answers to all the questions. And I still want to answer the questions coming up, but probably we have just limited time for today. So I think for those who posted questions and didn't get the live answer, I think we'll be posting the answers with some advices from Professor Lucas onward, and then we'll post these answers to our website so that you can look up those questions and answer together on our website. All right. Okay. Thank you very much for all of you who participated in this webinar. Thank you so much for this, participation in this webinar, and we expect from in order to continue our endeavor in discovering the new phases of the lighting effects closely related to the human biology. We will continue to share our ideas and technology through various channels like this webinar today. And we'll communicate with experts from different background and expertise. So in upcoming May, we'll return with the topic of relaxing lighting solution, which improves the quality of lives by providing better rest and sleep, as opposed to the energizing lighting solution that maximizes the value of space and help users achieve their goals in a more efficient way. So lastly, I'd like to ask you for your continued interest and support. Thank you very much for all of you. Thank you very much, Rob, for today's participation and today's valuable lecture. Thank you so much.
Thank you. Bye-bye.
Thank you. Bye-bye.